This video will explore the biosynthetic pathways for metabolic products of microorganisms. To effectively use microorganisms for economic purposes, we must understand their physiology, beginning with metabolic pathways. A metabolic pathway is a series of chemical reactions converting a metabolite into a final product. Anabolism forms complex substances, while catabolism breaks them down into less complex compounds. The compounds within a metabolic pathway are intermediates, and the final product is the end product. Catabolic reactions, often studied through glucose breakdown, provide energy as ATP and carbon skeletons for biosynthesis. Anabolic reactions build larger molecules, including cell constituents. 4 pathways for glucose breakdown to pyruvic acid are recognized. Industrial microbial products are broadly divided into primary and secondary metabolites. Primary metabolism involves reactions common to all living things, focusing on energy release and the synthesis of macromolecules necessary for survival. Primary metabolic products such as ethanol, lactic acid, and amino acids are associated with growth and peak during the logarithmic phase. Secondary metabolism has no apparent function for the organism's existence and is triggered by nutrient restrictions. Secondary metabolites are produced after the growth phase, in the late logarithmic and stationary phases. Secondary metabolism is restricted to certain species. Products often have unusual chemical structures, and multiple related metabolites may be produced. The ability to produce a secondary metabolite can be easily lost, a phenomenon called strain degeneration, potentially controlled by plasmids. Factors that trigger secondary metabolism, known as inducers, also often trigger morphological changes in the organism. Autoinducers of secondary metabolites include gamma-butyrolactones, N-acylhomoserine lactones, oligopeptides, and B factor. For example, the A factor, a butyrolactone, induces both morphological and chemical differentiation, such as streptomycin production in Streptomyces species. Microbial secondary metabolites are diverse, including antibiotics, pigments, toxins, enzyme inhibitors, and various drugs. These secondary metabolites profoundly affect health and economics. Their synthesis is complex, regulated by various factors, and often coded by clustered genes. The growth phases for organisms producing secondary metabolites are termed trophophase, the feeding and primary metabolism phase, and idiophase, where secondary metabolites are synthesized. Hypotheses for the role of secondary metabolites include the competition hypothesis, suggesting they help producers compete for food. The maintenance hypothesis suggests secondary metabolism sustains essential mechanisms when cell multiplication stops due to nutrient exhaustion. The unbalanced growth hypothesis proposes that over-synthesized primary metabolites are converted to secondary metabolites to prevent cell death. The detoxification hypothesis suggests secondary metabolites detoxify molecules accumulated in the cell. The regulatory hypothesis posits that secondary metabolite production regulates morphological changes, such as sporulation. The evolutionary reactions hypothesis views secondary metabolism as a testing ground for reactions that might eventually be incorporated into primary metabolism. Carbohydrates and increasingly hydrocarbons are the main sources of carbon and energy, fueling the catabolism that supplies carbon skeletons for both primary and secondary metabolites. 4 main pathways for carbohydrate catabolism to pyruvic acid are the Embden-Meyerhof-Parnas, the pentose phosphate, the Entner-Duderoff, and the phosphoketolase pathways. The Embden-Meyerhof-Parnas pathway reduces glucose to pyruvate. Aerobically, it feeds the tricarboxylic acid cycle. Anaerobically, it leads to various fermentation products. Diverse microorganisms ferment pyruvate into a variety of industrially important products. The pentose phosphate pathway provides no single end product, but a pool of various carbon phosphates. Its primary role is generating energy as NADPH2 and pentose for nucleotide synthesis. This diagram illustrates the pentose phosphate pathway. The Entner-Duderoff pathway is limited to specific bacteria and some fungi for glucose or gluconate metabolism. The phosphoketolase pathway is seen in bacteria, like Leuconostoc mesenteroides, where glucose and pentose fermentation yields lactic acid, ethanol, acetic acid, and carbon dioxide. Alkane catabolism involves oxidizing the terminal methyl group to an alcohol, then a fatty acid, which is broken down via beta oxidation, feeding acetyl coenzyme A into the tricarboxylic acid cycle. Alkenes, or unsaturated hydrocarbons, can be oxidized at the terminal methyl group or at the double bond, forming a diol before being converted and utilized as a fatty acid. Carbon flow in primary metabolism leads to catabolic products from pyruvic acid fermentation and anabolic products. Catabolic industrial products, such as ethanol, acetic acid, and lactic acid, are derived from pyruvic acid, with production heavily influenced by environmental conditions and organism species. Anabolic primary metabolites of industrial importance include amino acids, enzymes, citric acid, and nucleic acids. Secondary metabolite synthesis involves converting normal substrates into primary metabolic intermediates, assembling them unusually, and using special mechanisms related to general pathways, usually in response to non-growth conditions. Secondary metabolites utilize only a few biosynthetic pathways, which are linked to and use intermediates from primary metabolism. Secondary products can form from 1, 2, or more amino acids. Hidacidin, which inhibits plant tumors, is an example formed from one amino acid, glycine. Gliotoxin, an active antifungal and antibacterial, is derived from 2 amino acids. Secondary products from more than 2 amino acids include many important compounds, like toxins and peptide antibiotics.